We are really excited to be able to present this information to you today. It is part of research that has been funded by the Maine Shellfish Restoration and Resilience Fund of Maine. And we are going to be talking about Dr. Rob Holmberg's work to determine how acidic Maine mudflats are. And it's going to be showcasing results from monitoring mudflat chemistry in Beals, Maine. Dr. Rob Holmberg is a research scientist at the Downeast Institute, and he came to DEI from UMass Boston in 2019 as a postdoc tasked with designing and building an ocean acidification laboratory. And that laboratory is now completed and being used for research pertaining to ocean acidification and other stressors of climate change. So he's a research scientist studying OA and warming impacts on larval bivalves. And he, as you will learn today, also dabbles in carbonate chemistry monitoring on Maine mudflats. And with that, I will hand it over to Dr. Robert Holmberg.
Sarah, I'll share my screen here. All right, can everyone see that?
Yes.
Great. Thanks everyone for being here. As Sarah said, today I'm presenting the results from a project where we sought to create a long-term data set of mudflat porewater carbonate chemistry, which we observed on 2 different tidal mudflats in Beals, Maine. And we did this project with the generous support of the Broad Reach Fund. All right, so first I'd like to give a brief overview of ocean acidification. Atmospheric CO2 is currently on the rise due to anthropogenic activity and especially fossil fuel combustion. The red line is atmospheric CO2. This is warming the atmosphere in the process we call global warming. This is why, for example, the United Kingdom is currently on fire. It's also warming the surface ocean in the process we call ocean warming. This is why American lobsters are currently moving north out of Rhode Island and southern Massachusetts. But also a good deal of that surplus atmospheric CO2 is dissolving in the surface ocean, where it's shifting carbonate chemistry equilibria in such a way that seawater pH is reduced in the process we call ocean acidification. And seawater pH is the blue line. Right, and when I talk about carbonate chemistry, I'm really talking about 5 different parameters, of which pH is 1. The rest are total alkalinity, dissolved inorganic carbon, partial pressure of CO2, and the saturation state of aragonite, which we call omega. pH and omega are the carbonate chemistry parameters that are most relevant to the health and survival of marine organisms and especially calcifiers. But it's difficult to measure omega directly. So what we do is we measure any 2 of the other parameters and then calculate the rest of the carbonate chemistry system from those. Right, and omega is effectively a measure of the abundance of calcium carbonate minerals in seawater. Marine calcifiers like it when omega is high because it means they have plenty of substrate with which to build their shells. And they tend to not like it so much when omega is low. And so once again, ocean acidification is reducing seawater pH as well as seawater omega. All right, and bringing it back home to the Gulf of Maine, coastal systems like the Gulf of Maine are vulnerable to ocean acidification just like the rest of the ocean. However, the Gulf of Maine has certain characteristics that make it even more vulnerable. One of those is a good deal of freshwater riverine input from the Penobscot and Kennebec rivers. This brings fresh water into the Gulf of Maine and thereby diluting the buffering ions in the seawater. And also riverine input is increasing with precipitation due to climate change. Another source of acidification is eutrophication from wastewater effluent from city centers. And then we have a good deal of cold, low omega seawater entering at depth via the Labrador Current. And so as a result of all these things, seawater in the Gulf of Maine is expected to reach the critical threshold of 1.5 by the year 2050 in all regions. And Eastern Maine in particular, which is where DEI and the study sites are located, are expected to get there a little bit faster, just by nature of it being colder here. All right, so on the left, we have the carbonate chemistry equilibria that govern that system in seawater. And those same equilibria apply to the Porewater, that is the water between grains of sediment on tidal mudflats. However, Porewater tends to exhibit even lower pH and lower omega than the overlying seawater. And this is because of the influence of organic matter. So all the dead stuff and all the poop sink to the bottom where they get broken down by microbes in the sediment. And those microbes convert the carbon in the organic matter into CO2 that they exhale. And so even though the Porewater carbonate chemistry system is somewhat isolated from that of the overlying seawater, it's expected that ocean acidification is going to push it below its natural minima. And this could be cause for concern for things like clams that live in the mud. All right, so DEI has a long-running clam monitoring recruitment network project in which we put out these clam recruitment boxes on various mudflats all up and down the Maine coast from Wells all the way up to Sebayak. And the recruitment boxes are wooden frames with plastic mesh stretched across the top to keep out predators. And we put them down in the spring, and then we come back in the fall and count the number of clams that have settled in the boxes. And in Beals, we have two sites for the Clam Recruitment Monitoring Network project. One of them is a site called Perio Point on Beals Island, which is right on Moosabec Reach, which is the body of water between Beals Island and the mainland, Jonesport. And that's effectively open seawater. The other site is on Great Wass Island, which is connected to Beals Island, and it's called Squid Pond or Dobbins Island. And this site is in a cove adjacent to a lobster pound, and it's a little more closed off than the Perio Point site. And both sites have a total of 16 of the clam recruitment boxes lined up in the shore. So we thought it might be a good idea to start monitoring the carbonate chemistry adjacent to each of these clam recruitment boxes at our two Beals sites. So for the year 2021, my team and I have gone out and we've measured the pH and the temperature of the sediment pore water directly and then collected mud samples for later total alkalinity analysis. And this is really interesting because we looked in the literature and we failed to find any long-term data sets on pore water carbonate chemistry on tidal mudflats in the Gulf of Maine. There's really limited information on what the pH and what omega are like on these mudflats. And without having a long-term data set, it's really hard to know how climate change might impact these chemical conditions moving forward. So we sought to create the first long-term data set on this topic. All right, and this is a project that we are collaborating with the town of Beals and the Shellfish Committee in Beals. And specifically, we've been working with Butch Pinkham, who is the shellfish warden for Beals. And he's been coming out with us and helping us collect mud samples for carbonate chemistry analysis. Great, and I just want to take a minute to talk about the method that I'm using for measuring pore water alkalinity. I adapted Bob Burns' method for spectrophotometrically guided total alkalinity seawater titrations from the literature, and I chose it for its really high accuracy and precision. It has an accuracy and precision of 1 micromole per kilogram seawater, which is about as good as it gets. But what I found was that the authors of that method did not make their custom software for guiding the titration available. And also they used a totally different kind of spectrometer than I have in our lab. So I solved both problems by writing my own custom software for adapting the method to the spec that I have, which is the Agilent Carry 60 UV-Vis spectrophotometer. Beyond that, I also collaborated with DEI's facilities team to make a custom insert for this spec, which consists of a frame that holds an optical cell over the center of a magnetic stir plate and lines it up with the light path of the spec. And one of the challenges of working with pore water and analyzing the chemistry of pore water is that you don't have a lot of sample to work with. What we do is we go out and we take a 50 mil Falcon tube and then we scrape mud into the tube until it's full to the top. And then we take it back to the lab and centrifuge it to separate the pore water from the sediment. But even starting with 50 mils of mud, we usually end up with around 10 mils of pore water to work with, which isn't a lot. And this alkalinity method was developed with something more on the order of 300 mils of sample in mind. And so beyond writing the custom software and building our own equipment, we also scaled the method down so that we can use it with as little as 8 mils of pore water sample. And one of the solutions that I had to create to achieve that was creating a custom air diffuser. And that's what this looks like. And all that's doing is flushing the sample with nitrogen gas, but the diffuser is hook-shaped, so it shoots the bubbles up out of the way of the light path, which fires in between the top of that white frame and the bottom of the diffuser. And I'm happy to say that we're achieving the same accuracy and precision in the pore water samples that they did in the literature with larger seawater samples. All right, and so what we did was we went out once a month to both sites, Dobbins Island and Perio Point, throughout the entire clam growing season between April and November. And we measured pH directly using a handheld meter and probes. The pH probe was calibrated using a 1-point calibration to a tris buffer, which I made in the lab, and then in turn calibrated to the temperature that we observed out on the mudflat. So this is the result of the pH monitoring. In the figure, the white boxes are observations from Dobbins Island, and the gray boxes are observations from Perio Point. We have date on the x-axis and pH on the y-axis. And so this will give you an idea of how the Porewater pH changed at both sites over the clam growing season. So the CLDs, the little letters above the boxes, show the differences between months within each site. So we have two different CLDs. The black are for Dobbins Island and the red are for Perio Point. They're not comparing between sites, they're only comparing within sites. And then the stars at the top of the figure indicate where there were differences between sites within any given month. So for example, the pH at Perio Point was higher than Dobbins Island for the month of May, and also the months of July and August. All right, so overall, we observed a pH between 5.88 and 7.38 at Dobbins Island throughout the season, with an average of 6.8, and 73% of those observations were under a pH of 7, which is pretty low. At Perio Point, we observed pH between 6.07 and 7.59, with an average of 6.97, and 60% of those observations were below a pH of 7, which again is low. So overall, we observed, on average, a higher pH at Perio Point versus Dobbins Island. And as I mentioned before, there's really limited data on this topic in the literature, but two of the primary sources are from Mark Green and his work on flats in West Bath and South Portland. So for comparison, Green reported a Porewater pH between 7.03 and 7.05 on the Mudflat in West Bath, Maine, which was in May 2007. And our study sites were on average more acidified than Green's. And I just want to note that the pattern of pH change over the season at Perio Point is about what I would have expected to see, given what we know about the influence of phytoplankton blooms. So the primary producers are blooming in the spring, which brings the pH up because they're removing CO2 from the system. And then they continue growing through most of the summer until finally in the late summer and early fall, they die and then they sink, and then the carbon returns to the system and reduces the pH again. So we see sort of a bell curve in pH at Perio Point, which I think reflects that. The pattern of change at Dobbins Island is quite a bit different. So we see a reduction in pH over the summer before it stabilizes again in the fall. And I think this probably has to do with the mud itself at both sites. So again, Perio Point is more of an open ocean kind of site, whereas Dobbins Island is more enclosed. And at Dobbins Island, because of the rocks surrounding the mudflat, a lot of marine algae, macroalgae, gets trapped on the flat, which then decays and becomes part of the mud. And if you walk on both flats, you'll notice that the mud at Dobbins Island is a lot softer and stickier and smellier than the mud at Perio Point. And so I think what we're seeing here is a much greater influence of organic matter at Dobbins Island, which accounts for the unusual pattern in pH change. All right, so let's look at omega, which is the other parameter that's really important for bivalve condition. So at Dobbins Island, we observed an omega between 0.03 and 0.91. In other words, we observed 100% undersaturation with an average of 0.22. and 86% of our observations were less than 0.6. At Perio Point, we observed an omega between 0.03 and 2.87, with an average of 0.41, and 96% of our observations were below 0.6. And comparing that to Mark Green's data, he observed only 40% of his observations being below 0.6 in South Portland in 2009. And if you look at the figure, we see a similar pattern of change at both Perio Point and Dobbins Island relative to pH. So at Perio, we see a boost in omega in the spring and through the summer until it levels out again in late summer. At Dobbins Island, we see a bit of a curve, but overall, it's decreasing a little bit more through the summer than at Perio Point. And then the only between site difference I observed in omega was during the month of July. And during that month, the omega was a little bit higher at Perio Point versus Dobbins Island. And so I talked about the 0.6 threshold in omega. And the reason it's 0.6 is because, according to Mark Green's work, that's a really critical threshold in development for soft shell and hard shell clams. So Green et al. 2009 observed significant mortality of hard shell clams at an omega of 0.6. And then larger clams could take a little more stress. And he observed significant mortality in them at omega of 0.4. He also observed lesser soft shell clam recruitment in control sediments with an omega of 0.25 to 0.26 versus buffered sediments of 0.5 to 0.6. And finally, he observed hard shell clam soft dissolution at any omega under 1. And then in his 2013 paper, Green observed half the myo recruitment in control sediments, averaging an omega of 0.68 versus buffered sediments with an average of 1.3. And so that 0.6 threshold appears to be really important for dissolution mortality in settling clams. And once again, most of our observations at Dobbins Island were under that threshold. And even more of our observations at Perio Point were below that threshold, which indicates that settling clams might have a hard time in the environments at Perio Point and Dobbins Island. All right, so in summary, our study sites were on average substantially more undersaturated with respect to aragonite than the South Portland site. Green studied, and the carbonate chemistry conditions at our sites were consistently below Green's thresholds of biological disruption, which might indicate that Dobbins Island and Perio Point are chemically hostile environments for juvenile soft and hard shell clams. And it's likely that some of the settlers are dying due to dissolution mortality. And once again, I just want to underscore the importance of measuring both parameters, because if we had only measured pH, like a number of other research teams, we wouldn't be getting the full picture of carbonate chemistry on these flats. All right, and so once again, we were measuring these conditions adjacent to clam recruitment boxes, so I'd be remiss if I didn't talk about what we found in those boxes in terms of clam abundance at both sites. So for the year of 2021, the average clam density was higher at Perio Point versus Dobbins Island. And once again, the carbonate chemistry conditions were more favorable for calcifiers at Perio Point versus Dobbins Island. So at first glance, it appears to be possible that we observed more clams at Perio because of the more favorable carbonate chemistry conditions. And then again, we also observed more clam recruitment at Perio Point versus Dobbins Island, which may also be due to the more favorable carbonate chemistry conditions. But there's one detail that I've glossed over until now, and that is the influence of green crab predation. And so we observed more green crabs at Dobbins Island versus Perio Point, and those green crabs are eating the young clam recruits. And so it's likely that at present, green crab predation is a more important vector of mortality in these young clams than carbonate chemistry. But moving into the future, as the conditions worsen, it's likely that the clams are going to be facing the double whammy of green crab predation and hostile carbonate chemistry conditions. And our future work is going to be about trying to tease apart the relative importance of those stressors. All right, so those were the results from the year 2021 clam growing season, but we did secure funding for a second year of monitoring, which is currently ongoing. Once again, we started in April, and we've been going out once a month to the same sites and taking our measurements and samples. And the overarching goal here is to create a long-term data set over several years to determine whether these conditions are changing over time with climate change and give us something with which we can maybe anticipate how they'll change further in the future. So before I take questions, I just want to plug the next DEI climate change seminars for the year. Wednesday, August 10th, Sarah Randall is going to be telling you more about the green clam problem in Maine and its relevance to the clam monitoring and recruitment network. And then on Tuesday, August 16th, our research fellow, Ray Chaya from Stony Brook, is going to talk to you about his meta-analysis regarding upwelling exposure to bivalves. All right, and with that, I'll take questions.
Thank you, Rob. And so if someone has a question, you can raise your hand or put it in the chat.
We have a question here in the conference room.
Awesome.
Sure.
Hi, Rob. Can you hear me?
Yes.
Cool. Okay, so you got the sediment or Porewater pH more than not being below 7.0. Presumably, you'll have water column pH being more than not above 7.5. And so what I'm wondering about is that like benthic boundary layer, that 1 inch or 1 centimeter right above the sediment where perhaps the siphons of the clams are interacting with. And so that benthic boundary layer may be very physiologically important for the animals. Where in that pH range do you suspect that benthic boundary layer pH lies?
That's a really good question. And specifically, I was measuring the pH and the omega in the upper half centimeter of sediment. And Ray's talking about the layer of overlying water right above that. I guess I'd have to find a creative way of measuring that in between tides. I would expect it to be somewhere in between.
We have a question in the chat from Mike Johnson. He says, great presentation. You found the green crab presence and clam mortality higher at Dobbins. Do you have any insight on why that may be?
Sarah, would you like to take that one?
Yeah, I mean, it's a matter, yeah, I don't know. I mean, that is something that we're trying to figure out with the long-term monitoring that we're doing with the Clam Recruitment Monitoring Network. And so far, we are on our, we're on our third year of data collection on this. And of course, it's really important to have many years in order to identify trends and see if those are consistent year to year, along with the levels of clam recruitment. So, no.
I'll offer one hypothesis, which is untested, which is that I mentioned that Dobbins Island is more enclosed and it tends to trap macroalgae. So it's possible that the green crabs are using the macroalgae as a shelter.
And that sounds like a good hypothesis, because we know that the presence of habitat is a key factor in crab presence, especially the small ones that are eating the clams. Do you want to speak about the where in the mudflat you were sampling and how that might be, that could, I don't know, the influence of the pH, at least I know, can be determined by like where in the tidal gradient you're sampling?
Sure. So we set up the recruitment boxes at the 0 line in the tide. So they're I'd say they're about, at least at Dobbins Island, they're maybe 30 feet from the high tide line. And we set them up parallel to the tide. So we're capturing one specific horizontal line on each mudflat. In terms of how the carbonate chemistry conditions might be different at different distances on the flat, I don't really know. There's no data on it. But I can tell you that there's a huge gradient in pH and omega at depth within the mud. And so we try to standardize our measurements to a specific depth for everything. And that's the depth where the young clams are going to be settling.
And the young clams are more susceptible to effects of OA, right, Rob? Than like an adult.
Yeah, so in general, the younger life stages of marine calcifiers tend to be more sensitive to pH and omega stress than older life stages.
Carolina?
Thank you, Sarah. Thank you, Rob, for your presentation. Great work. Hey, Rob, you have a lot of variation among samples, which is typical, right, of ecological samples and chemistry in the space of the sediment. So,
Sure.
how many samples, like, do you think there would be a point in the number of samples where we can lower that variance or that variability to make it more clear the trend through time and between sites? And I guess that my second question will be, you have lower values than the previous work in 2007, and I guess it's premature to think about that those changes have to occur through time, so we need more data to support that, right? But would you think that there would be differences between these sites, the West Path and your site, that could explain also the difference in the chemistry that you're finding, like something like related to temperature or salinity or other factors that doesn't necessarily mean a trend through time?
Yeah, sure. So to answer your first question, at each site we sampled from beneath each of the 16 recruitment boxes each month. And so we have 16 samples of pH and 16 samples of omega from each site within each month. And there's, I consider that a pretty good number of samples. In one of the green papers, the sample size was something like 3. And so we have a lot of observations. And certainly I think having more observations would reduce the variation that we're seeing. In terms of practicality, 16 samples is about as much as we can get through during low tide before the tide comes back in. If I had a larger team, maybe I could go out with more people and collect more. And then to answer your second question, I have to assume that there's some influence of latitude here. So we're a bit further north and a bit further east than West Bath and Portland. It's a little bit colder up here, and both CO2 and calcium carbonate are more soluble in colder water. And to expand on that, I have to assume that the population of clams that live up here are a little more adapted to lower pH and more undersaturated pore water conditions than the clams further south. Though I don't have any evidence to prove that.
Thank you so much. Keep your great work.
Right, we have another question from the chat. It says your samples were taken at low tide and thus sampling water that may be released from the sediments. Do you think that this would differ when the tide is in and there could be more water exchange between the water and the sediments and the overlying water?
Yeah, certainly. And, one of the things that I've been thinking about is, which would solve both Carolina's question and this question is, can we set up some kind of continuous monitoring station, at least for pH in the pore water? There's been some work on pore water wells. We could maybe create a pore water well and, stick a box in it that measures pH and maybe collects samples intermittently for alkalinity or DIC or whatever. That way we'd have information from, not only low tide versus high tide, but also how the conditions change throughout a low tide cycle. Are the conditions that I measure in the first box of low tide different than the conditions I measure in the final box just because they've had that much more air exposure? So certainly things I'm thinking about.
Right. Any other questions for Rob? Rob, anything else you want to get out there about your work before we close? We can close a little bit early if there's no more questions.
Nothing in particular. Last week I presented the results of a manipulative pore water study in which we placed different sizes and densities of shell hash on a mudflat in South Portland, the same mudflat that Mark Green worked on, to determine whether there was some magic combination of size and density that is most effective at buffering pore water pH, and also whether that has any impact on clam abundance on the flat. Beyond that, I have a number of ongoing ocean acidification exposure trials in the lab at DEI. We're currently working on a blue mussel project for sea grant, in which we are exposing larval blue mussels to different combinations of pH and temp, and also different microalgae diet treatments to see if we can optimize their diet in such a way to make them more resilient to climate stress in a hatchery scenario. And for those projects, we're actually settling the mussels onto ropes in my experimental tanks and then deploying the ropes in the ocean at mussel farms. And that's my update.
Thank you. Thank you, Rob, for sharing today. And I would like to thank everyone for joining us. This video, this seminar will be available on YouTube shortly. And please join us next week where you can listen to me talk about green crabs on Wednesday, August 10th at noon. So thanks, everyone.